Well, thanks, Mateen, and thanks very much to the organizers, Fritz and the rest of them. I'm sure they're not here because they probably have a more important session to go to. It's a great honor to be asked to give the state-of-the-art lecture. So I was asked to talk about the serpins. They wanted me to talk about Protease nexin 1, and I will talk about that at the end. But I thought I'd give an overall review of serpins that inhibit my favorite protease in the blood coagulation system, which is thrombin. Now, I do have just one little thing to declare, and that is I do have an affiliation with a company called XO1, but it's not really related to what I'm talking about today. So first, a little introduction to what I like to call thrombin's life cycle. It's generated on the subendothelial membrane when tissue factor and factor 7A come together, and they make a little bit of 10A. 10A makes a little bit of thrombin, and this thrombin can then bind to platelets that are in the vicinity, and it binds via GP1B alpha. And that thrombin then can cleave PAR1 much more efficiently. And so when you reach a certain threshold of thrombin, then you get good activation of platelets, and that flips the platelet membrane so that you now have phosphatidylserine on the outside, and that supports the assembly of the binding of factors 8 and 5, and those are cleaved and activated by thrombin. And then thrombin can bind and cleave to other PARs. And then we get the assembly of what I like to call the engines of blood coagulation, and that's the 10-ACE complex over here, and the prothrombinase complex, and essentially they are there to make a lot of thrombin. And this thrombin can then go and feed back to activate more platelets, to cleave fibrinogen, and of course to feed back to increase its own formation. And there are a couple talks that I want to draw your attention to. I'm actually giving a talk later this afternoon on the behalf of a graduate student or former graduate student, Bernard Lechtenberg, showing that GP1B alpha, how it interacts with thrombin. And there is a talk also by a postdoc in my group. He solved the structure of the Pseudonaya textilis prothrombinase complex, and that's actually a very interesting structure, a very interesting talk. At the last phase of the thrombin life cycle, it actually turns itself off, and it does this in a couple active ways. One, it binds to thrombomodulin over here, and this is through EGF domains 5 and 6, but also if there's a chondroitin sulfate moiety available, it will bind also via exocyte 2 to the chondroitin sulfate. And it activates protein C, and activated protein C goes and then shuts down thrombin generation by cleavage of factor 5A and 8A. But it also plays an active role in recognizing its endogenous inhibitors, and these are serpins, and this is now shown on glycosaminoglycans here. And these serpins then will essentially crush the thrombin, as I'll show you later, and it allows it to be removed from whatever cofactor interaction it's on, and then it gets cleared by binding to LRP in the liver. And we have some nice unpublished data on that, but we won't be talking about it at this meeting. There is another talk, actually, that I'm going to draw your attention to by a graduate student in my group who's talking, this is Stephanie Polderdijk, who's going to be talking about how protein C inhibitor inhibits both thrombin and APC in a manner that's dependent on the size of its reactive central loop. And a really interesting talk by Jonathan Langdown, who is showing a rare bleeding disorder from a family that have a truncation in the thrombomodulin gene so that it actually gets shed, and that leads to an increase in bleeding in these people. So that's thrombin's life cycle. This is thrombin's structure. Now, anytime you see thrombin or any serine protease of this family, this is the chymotrypsin family of serine proteases, they should be in this orientation. This is the standard orientation. And this is colored according to its from N to C terminus, so from blue to red. And some of the more important features here are shown. We have the catalytic triad here in the middle, the oxyanion hole here, and the sodium binding site over here. In this orientation, which is the orientation I'll normally show for thrombin, we have exocyte 1, which is over here between the 30s and the 70s loop. And we have exocyte 2, which is over here around the back. And substrates are going to be binding from over here, from the N terminus, to over here on the C terminus. And you can see that in this representation here. This is a electrostatic surface representation of thrombin. This is with a reactive central loop of heparin cofactor 2 in there. And blue is, in this case, positive, and red is negative. And you can see that it's a rather deep active site cleft. And it binds extensively these reactive central loop peptide regions from the serpins. And if you rotate this 45 degrees in this direction, so this is on the P prime side here, then you can see this very positively charged exocyte 1. This is actually really close to the active site, and so it's really where the P prime part of a substrate peptide will be coming out, and that's how actually PAR1 recognizes it. This site also binds the E domain of fibrinogen, and it also binds to thrombomodulin. And this is also, even though it's a basic site, it's also highly hydrophobic, and so most of the energy of the binding is provided by hydrophobic interactions. So if we rotate it back to the middle position again, and then rotate it 65 degrees in the opposite direction, you can see around the back of the molecule is a very basic, this is the heparin binding site, or exosite 2, and this site is responsible for binding to certain things like this is the fibrinogen gamma prime region. It also binds to heparin, this is a structure that we solved, and it also binds to GP1B alpha, and as you'll hear later if you come see my second talk, it exclusively binds to exosite 2. So that's thrombin introduced, and now what are serpins? Well, serpins are serine protease inhibitors, but that's just really an acronym for the family. They have a core domain that's about 400 amino acids in length, and there are well over 1,000 identified now. There are 36 serpins in the human genome, and they're found in all types of organisms from bacteria all the way up to us humans. And what makes them very important and very interesting and relevant for this meeting is that they're involved in lots of processes that are tightly regulated, in particular blood coagulation, fibrinolysis, complement, and inflammation. And of most importance for this meeting really is the serpins involved in blood coagulation. So this is the standard orientation of a serpin, and this is again colored from the N-terminus in blue to its C-terminus in red. And it really is a single domain, but it kind of is like a two-domain molecule with a helical domain on the N-terminus and a beta-barrel domain on the C-terminus. But this is the way we normally color and look at a serpin, and it has its two main features highlighted. So it's got a beta sheet A, which is in red, and it's 5-stranded, and that's one of the main features of the molecule. And then it has a 20 amino acid reactive central loop here that starts here at about P17 and goes to P3. And this nomenclature is a substrate nomenclature so that this P1 prime bond is the bond that's targeted by a protease, so the sisal bond. And then we number the residues going this way just sequentially, so from P1 all the way to P17 here. And we number them on the prime side, on the C-terminal side, from P1 to P3 prime. And this region here that's circled is not overly relevant for this particular talk, but that's called the hinge region. Now, these strands are numbered 1 to 6 here, and we left out strand 4, and the reason for that is that the first structure of a serpin ever solved was that of cleaved antitrypsin, and you can see that the P1 and P1 prime bond has been cleaved, and there's a 70 angstrom distance between the two, and that's why it's incorporated from about P15 down to P3 as the fourth strand of the now 6-stranded beta sheet A. This is a really important conformational change from its mechanistic point of view, but it's also really interesting in that this conformational change increases the stability of the serpin by over twofold. So it goes from being a protein that will unfold at about 55 degrees Celsius to over 120 degrees. You can't actually unfold these things unless you add SDS and boil them. And the best estimate for the energy that you get out of this conformational change is about 32 kilocalories per mole. I mean, it's a lot. So this is how serpins inhibit serine proteases. This is based on the structure that we solved and published in 2000. And I'll just show it again because we have to make these movies play automatically. The protease comes and it recognizes the sisal bond. It recognizes that as a substrate, and then it actually cleaves the bond. But because the serine, the catalytic serine, is covalently linked to the substrate, in this case the P1 residue, it's covalently linked for all steps throughout the catalytic cycle until the deacylation step at the end. At that step, or before that step actually takes place, the protease is actually translocated to the opposite pole of the serpin, and it's destroyed. And you can see the flashing bit here is about 40% of the protease that we couldn't see in the electron density of the original structure. So this is the serpin mechanism in a nutshell. You get the protease that attacks this bond here at the top, and before it can finish the catalytic cycle, it's actually translocated to the other end and destroyed. And so what happens to thrombin when it's inhibited by serpins? Well, now this is the standard orientation of thrombin now, but colored according to hydrophobics. And you can see here exosite 1 and exosite 2. Now, if I made a model, I did make a model of thrombin based on the structure of the final serpin protease complex that I showed you a second ago. And that's the part of the structure that it disappears. So exosite 1 essentially disappears. It becomes conformationally disordered. And in fact, there were two papers, one that came out before our structure and one that came out afterwards, that actually showed that exosite 1 is actually functionally disordered and destroyed when inhibited by serpins. And then in 2010, we also showed that exocyte 2 is also functionally disordered and destroyed in complex with serpins. So this is important because, of course, thrombin is going to be bound to cofactors on whatever membranes so it can be bound to the fibrinogen, and this would release it from fibrinogen. It could be bound to thrombomodulin. This would release it from that. It could be bound to GP1B alpha. It would be released from that. It could be bound to heparin. It would be released from that. So this is important, really, so that you not only get inhibition of the protease, but you get destruction of the exocytes, so that it gets released from whatever it may be bound to. And this is important, and this is why serpins are the only endogenous inhibitors of thrombin. So, like I said, this is the mechanism in a nutshell, but the rest of my talk is about the central bit, which is recognition. So the protease has to recognize the serpin as a substrate. And this is, of course, the reactive central loop composition is very important, but there are also exocytes that are critical. And we've solved the structure of, I think, most of the relevant thrombin-serpin complexes, and I'll just touch on those now. So they are antithrombin, or AT, heparin cofactor 2, or HC2, PCI, which is protein C inhibitor, and a bit of a misnomer for that. And there's protease nexin 1, which is a fabulously good thrombin inhibitor. And I just sort of briefly wrote down where I think these inhibitors are actually inhibiting thrombin. And I think the principal inhibitor of thrombin in the lumen, so when there is no cofactor involved, is antithrombin. And I think the best evidence for that is that antithrombin levels, when they're low, that essentially predisposes to thrombosis. And that doesn't make sense if it's the heparin-activated form that is the functional form. And the reasons for that I won't go into. But heparin cofactor 2 and antithrombin work on glycosaminoglycans. Antithrombin will be working on the heparan sulfates, and heparin cofactor 2 on the dermatan sulfate. PCI and protease nexin 1 are also GAG-dependent inhibitors of thrombin. Protease nexin 1 is interesting because it's actually secreted, probably pre-bound to aglycosaminoglycans. So it actually never leaves the cell on which it's expressed, and it stays on the cell wall for its entire life. So we also have the vessel wall interaction with thrombomodulin. Probably the principal inhibitor of thrombin-modulin-bound thrombin is protein C inhibitor. And of course, protease nexin 1 is an important inhibitor on the surface of platelets, and it shows that another group actually has shown that its presence there helps set the threshold of thrombin concentration for platelet activation. And it's an incredibly good inhibitor of thrombin. So there have been 5 papers that have been published and 6 structures in total, and these are summarized in this table. All of them, save for one, were solved by my group, and there are 2 of independent structures of antithrombin, these 2 at the top, and that's in the presence of a heparin-like molecule. Then there's one of protein C inhibitor with heparin, although we couldn't see the heparin in that one, and heparin cofactor 2. And then we have 2 independent copies of protease nexin 1 in the asymmetric unit in a single crystal. So in all, we have 6 structures of thrombin bound to serpins in this initial Michaelis complex, so the recognition complex. And I like to break down how they're interacting with the serpin based on the number of residues, as you can see here. And then we have what I like to call the reactive center loop and the exocyte interactions. And reactive center loop is everything from P4 to P3 prime, and exocyte is everything else. And you can see that this, in terms of total buried surface area for the complex here, for what's contributed by the reactive center loop and what's contributed by exocytes. So anyway, we'll deal with these individually as we go on. But before we do that, this is what all the structures look like if you superimpose the thrombin component. And you can see that, so essentially the active site is facing down, and the serpins are going to be attacking going up. And you can see that there are many different ways that you can approach the active site because the reactive central loop is so flexible. This is now with the serpin component superimposed, and again, you can see that there's a lot of variability here, and that you can have interactions that are towards the front of the serpin here, or all the way towards the back, as you can see over here. So there's a lot of room, and that's because the reactive central loop is flexible, and because exocytes need to be engaged in some cases along the top surface of the serpin. So this is now looking down into the active site cleft of thrombin, and we're just throwing in P6 to P4 prime of this is antithrombin. This is the second structure of antithrombin. You can see it's quite different. Here's heparin cofactor 2. Here's protein C inhibitor. And here's the 2 protease nexin 1s one that's in red, and the other one, which you will see is very strange indeed, which actually comes out prematurely, and I'll talk about that one later. But except for the magenta guy here, which is the second structure of Protease nexin 1, the only thing that's conserved in terms of the interactions are the P1 and the P1 prime positions, and that's because they have to be aligned perfectly in order for proteolysis to commence. So there's a lot of flexibility as to how the reactive center loops can be recognized. And in fact, for a lot of serpins, I would say for the majority of serpins that are regulated, the reactive center loop sequence is not optimal for the protease that's its real target. The reason for that is that you want to be able to regulate its binding with cofactors and exocytes. So these are the two structures of antithrombin bound to thrombin. So this is the one that we solved here on the left, this 1TB6, and you can see that we have the entire heparin moiety, which actually bridges the two. And this other one here on the right is from Peter Gettin's group. And you can see they're very different, in fact. A couple of differences that are clear is that we had full density for the entire heparin hexadecasaccharide, and they didn't. The other thing that's quite interesting about it is that we have a forward orientation of thrombin, and that lines up the heparin binding site, exocyte 2 of thrombin, with the heparin binding site of antithrombin. And there's a 46-degree rotation and an 11-degree translation of the thrombin, so the heparin binding site is actually around towards the back. This structure here that we solved is actually consistent with the fact that loop expulsion of the hinge is not important for thrombin being bridged by heparin. The thing about their structure also is that because the heparin binding site's been rotated around back, it's too far to really be bridged by a 16 sugar or chain sugar. You would need 2 more sugars in order to bridge it. So we think that both are obviously real structures. Both are potentially physiologically relevant, but this one here is more consistent with what we know about the bridging mechanism. So this is now the structure of thrombin, just colored according to interaction with antithrombin. This is the 1TB6 and this is the 1SR5 structure. And you can see the major difference here is the engagement of this gamma loop right here, or the autolysis loop, which we have in our structure, and they didn't in theirs. And there's a large difference in the amount of buried surface area. So this is heparin cofactor 2, and I've talked about heparin cofactor 2 a couple times, and so I'm not going to go into any details about that, but this is an incredibly interesting serpin, and it interacts via exocyte 1, as you can see here, using this 80 amino acid N-terminal extension, and it interacts very much in a hirudin-like fashion, and this attracts it to bind to the reactive center loop here, and it has a leucine at the P1 residue, and like I said before, the serpins that are inhibiting something that is highly regulated and that uses cofactors tend to have a non-optimal reactive center loop sequences. This has a leucine at the P1 and not an arginine, which every other substrate of thrombin has. But anyway, I won't go into this in any more detail, but just to look at how much of the interaction interface is by exocytes, really astounding. This is protein C inhibitor, and this actually does not have a lot of exocyte interaction, and it has a very long reactive center loop, so it can kind of hold thrombin at bay. It can keep thrombin away from the surface of, and especially if you compare this intimate contact with this one, you can see that it's actually keeping thrombin away. The only real interaction here is with the 60s loop, as you can see there. And this is the last one I'm going to talk about, which is Protease nexin 1. And this is really interesting, actually, because, as I mentioned before, we had 2 complexes in the asymmetric unit. And normally, if this happens, you would expect them to be identical. But they're not. They're very, very different. This is complex 1, and this is complex 2. And you can see that complex 1 is engaging the P1 residue, and complex 2 is engaging the P1 residue, but in a totally different manner. You can see that there's a huge rotation also in thrombin going from here to here. Now, what's interesting about this is that protease nexin 1 is always bound to glycosaminoglycans. It has the same heparin binding site on helix D, which is around the back there, as antithrombin does. And the heparin binding site is essentially this region right here, which is aligned in this structure, and it's way back here, so not aligned in the second complex. And even more interesting than that, if you look at the reactive center loop interactions, we have this is complex 1 in cyan and complex 2 is in yellow, and you can see that complex 1 actually doesn't engage the P1 prime residue. This is the P1 arginine, which actually does go into the S1 pocket, but the P1 prime residue is over here, not engaged in a productive fashion. So it's not a productive Michaelis complex. It's not ready to be cleaved. But it is still a complex, and it is still engaging thrombin in a fashion that buries the P1 residue into the S1 pocket. But interestingly, if you just put a heparin molecule in there, you align here the heparin binding site of protease nexin 1 with the heparin binding site shown by balls here. In complex 1, they're aligned. In complex 2, they're misaligned. So in other words, it looks like this non-productive complex here is the initial complex when thrombin and protease nexin 1 initially encounter one another, but they haven't yet engaged the reactive central loop in a productive fashion. This could be an inhibited thrombin. This could be part of the mechanism by which thrombin is inhibited by protease nexin 1. I don't know how long this complex lives for, but it will be an inhibitory complex. Then, at some point, the thrombin rotates around, it moves its exosite 2, so its heparin binding site, away from the linear heparan sulfate chain and fully engages the reactive central loop so that proteolysis can begin. Now, I've made a little movie to show this, and I'll show you that in a second. But before I get to that, one of the most interesting observations from this structure is that this is the complex. This is now colored according to B factor or temperature factor. So what this is it's an indication of the structural stability. If it's blue, it's stable. If it's red, it's unstable. And what we have in complex 1, which hasn't productively engaged the reactive central loop, we have a very stable thrombin. In complex 2, which has productively engaged the reactive central loop, we have a very destabilized molecule. Now this is interesting because if you look at the region that's been destabilized, it's actually exosite 2, indicating that this would be deficient in heparin binding, even though it's engaged the reactive central loop properly. So this one would be bridged by heparin, this one would not be bridged by heparin, and we have a destruction, a conformational disordering of exosite 2. So this is how we hypothesize that thrombin is inhibited by protease nexin 1. We have an initial encounter complex, which aligns the heparin binding sites of thrombin and protease nexin 1, this beautifully positive charged region here. And this complex could live for a while. It is an inhibited complex because, of course, it's engaged non-productively and so cannot be cleaved. But then we get a rotation, full engagement of the exocytes and disengaging of the heparin from the exosite 2 of thrombin, and a conformational disordering of exosite 2. And this really does set the stage now for reactive center loop insertion and full inhibition of thrombin. So to conclude, it's clear that thrombin is a very, very important, I would call it a pleiotropic protease. It has multiple functions and multiple inhibitors because thrombin finds itself bound to different things on different cell types, and because of that, you need different serpins. Thrombin recognition is mediated by exocytes and obviously by cofactors, and thrombin is functionally destroyed by serpins, and that is essentially why serpins are the only endogenous inhibitors of the important serine protease, thrombin. And just to acknowledge people that were involved, in particular Lee Wei, who was a great postdoc with me for many years, now has an independent position as lecturer at the University of Cambridge. And she did really most of the work that I talked about today. And then collaborators who have been really helpful for various stages of different projects. And thank you for your attention.